Yes.
Okay, thank you. So, wherever you're ready, Kaavya?
Okay. Can you able to see my slides?
Yes, but I think you must put on the, because we see also your
That is because you're using two screens.
yeah,
Okay.
we're in your notes.
Okay.
Okay, thank you.
Yeah. Thank you. A very good morning to everyone who's attending this presentation. I really appreciate this opportunity and I'm really glad to be a part of this workshop. I'm Kaavya, a PhD student working under the supervision of Dr. Sergio Martinez. I'm working on a transdisciplinary work which includes some part of dairy manufacturing, engineering, and chemistry. And today I'm really excited to talk about my research work on extracting the valuable phospholipids which goes as a waste using new type of smart solvents that are switchable. So moving on to today's contents, I'll be talking briefly on the importance of phospholipids, what are the existing techniques which are present for manufacturing of the phospholipids and the challenges and how the current approach used to mitigate these challenges. And I'll be talking briefly on the materials and methods used in this research, and I'll be emphasizing very interesting results from these 4 different studies, and I'll be talking on the few conclusions and also how this research is going more forward in the research. Going on to the importance of phospholipids. Phospholipids are lipid bilayers found in all plant and animal cell membrane. They refer to a class of complex polar lipids which consists of a phosphate group and 2 fatty acids esterified to a glycerol backbone. They have a polar head and a non-polar tail, which is like the amphiphilic in nature that helps in the emulsions and everything. So in case of the milk, the total milk fat is the phospholipid account for about 0.5 to 1% of the total milk fat, depending on the season and lactation stage. And they are located in the milk fat globular membrane in arrangement with the membrane proteins. Such arrangements has caused the milk fat to be emulsified and dispersed within the milk. Why these milk phospholipids are very relevant and why they are also considered very unique compared to the vegetable source such as the soy and the egg yolk is because of the relatively high concentration of the sphingomyelin and phosphatidylserine which is present in the milk source, and they have a lot of functionality as well as nutritional and health benefits. For instance, consuming these phospholipids helped in reducing cardiovascular disease, cholesterol absorption. In recent times, it is used in the infant formula for the growth and brain development of the infants, and is also used in various processing aids for value addition as well as a natural emulsifier. So what is the current challenges, not only in the dairy processing, the soybean processing, the algae research, all are facing challenges in extracting these phospholipids. Since it is a very multi-step procedure which involves like concentration, fractionation, distillation, and they use a lot of chlorinated and flammable solvents which are injurious to health, and also they are results in low extraction efficiency, and also it is economically unviable. So in the dairy processing, since it is a very wet dilute material, this is a simplified diagram of an isolation process of phospholipids. Since it is a very wet dilute material, it has to be concentrated and mixed with various solvents and then centrifuged, and it has to be distillated. The solvent has to be distillated, evaporated, and then you get a fraction, small fraction, phospholipid fraction. So what the industry which is in use, they use a concentration process. So a byproduct is taken and then they use some proteolysis to disrupt the protein matrix, and then they go through some filtration techniques such as microfiltration or the ultrafiltration, where they come out with the return tate. The return tate is further evaporated, dried, and it has a concentrated form. And these studies show um, also show that the microfiltration in combination with other supercritical carbon dioxide techniques, and also some sequential techniques which involves ethanol and renewable alcoholic solvents, all these resulted in around 19 to 58 percentage of phospholipids. And commercially available phospholipids are from the Fonterra and the Ala foods, which are usually used in the infant formulas. So all these have led to an enriched form of matrix rather than extracting from the byproduct by itself. So we came up with an alternative approach to is the use of switchable solvents. In this case, these switchable solvents are made of either primary, secondary, or tertiary amines without any kind of pre-treatment. We just use the matrix by itself, a wet, very wet dilute material. And the switchable solvent speciality is based on the reversible reaction of an amine, which changes its hydrophilicity from water immiscible form to water miscible form by simply bubbling it with carbon dioxide. When you remove the carbon dioxide, it goes back to its original form. This is the whole speciality of the switchable solvents. And the change in miscibility is because of an acid-base reaction between either the hydrated carbon dioxide or the carbonic acid in the carbonated water and the switchable solvents, which results in an hydrophilic ammonium bicarbonate salt. So which is hydrophilic in nature. So when you remove the carbon dioxide or you flush in the nitrogen, it goes back to its original form of amine, so that you can reuse the solvent for further extraction process. In that way, it's a very sustainable technique. In our research, we used N,N-dimethyl cyclohexylamine into toward the process. So the overall hypothesis of my work is to use switchable solvents as a suitable technique for the phospholipids from the dairy byproducts. And these four are the very specific objectives which I'll be talking on today, on my today's work. So moving on to the materials and methods. So this amine was taken for three different byproducts, which is bee serum, which is a byproduct from anhydrous milk fat, buttermilk, concentrated buttermilk were used, and the switchable solvent were compared with the traditional Falch extraction method and various analytical methods, particle size, zeta potential, microscopic structures like the scanning electron microscope, confocal laser scanning images, HPLC were utilized in all the four different studies, but then I'll be highlighting very few interesting from each study. So this is a schematic diagram of how a switchable solvent extraction is carried out, a byproduct along with the switchable solvent ratio. For example, 1 gram of the dairy byproduct, either bee serum or buttermilk, along with some switchable solvents like 3, 6, or 12 ml dependently. The solvent is being kept for stirring, and then equal volume of water that of the solvent was added. This was done in order to maintain the stoichiometry of the reaction. If not, water is not added, it can't convert to its salt form. So that is a critical step. And then it was left for carbon dioxide, you can see in the step 3. And when you remove the carbon dioxide, you can see a layer of lipids floating on the top. And then further with the solid phase extraction, we obtained the phospholipids. So in this case, we added 1 ml of hexane to facilitate the extraction of the lipids. But then in case of when we do it in a large scale, there is not necessarily hexane has to be added. And this is an ultrasoundic pretreatment which is used for the study 2, where 3 different acoustic intensities were used as a pretreatment for time. And then these are the 4 in my third study of using the extraction conditions for the bee serum, 27 experimental trials were carried out for the 3 independent variables: temperature, solvent ratio, and time. Moving into the most interesting part, the results. moving on, my first study, we evaluated the use of switchable solvents, the N dimethyl cyclohexylamine, for the 3 different dairy products: bee serum, freeze-dried buttermilk, and the buttermilk. And we were able to obtain very promising results, and it was observed that the switchable solvent ratio gave a higher yield of phospholipids in all the 3 dairy byproducts. While I explain, I would like to gain your attention at this point, where we were able to obtain 99% of phospholipids in the buttermilk, which was nearly 4-fold higher compared to the traditional fudge extraction method. So however, in case the switchable solvent extraction had an influence on the type of the byproduct used, in case of the freeze-dried buttermilk, it was around 77% of the phospholipids. In bee serum, it was around 7.5% of the phospholipids. So this switchable solvent behavior, which has a very low volatility behavior, helped in condensing the layer of the fat, and this mechanism of the switchable solvent will be discussed in the later slides under the mechanism study, how the switchable solvent worked to give a higher yield of the phospholipids. So this is a product distribution which was quantified using a HPLC, using charge aerosol as a detector, which shows there were 5 different individual phospholipids were detected, like the PI, PS, PE, and PC, and it clearly outweighed the Fulch extraction method. And this particular compound, sphingomyelin and the phosphatidylcholine, has a relatively high proportion, and it has significant in the infant formulas because of its nutritional benefits. Further, we were working on the bee serum matrix because it has a potential interest in the dairy industries, and in order to improve the extraction efficiency, ultrasound pretreatment was applied. This was done since these ultrasound generates sound waves that creates some rarefactions and compressions, creating a turbulence, and it helps to reduce the size of the flat globules, and also helps in, by improving the mass transfer, it helps in separation of the phospholipids. It was observed that the acoustic intensity 44 gave a higher yield of phospholipids in all the switchable solvent ratios as well as the Fulch extraction. It was interesting to note that this acoustic intensity 44 with the switchable solvent ratio 12 gave a higher yield, which was around 70% of the phospholipids, which was 10 times higher compared to the untreated samples. And in case of the individual phospholipids detected using HPLC, it was clearly observed the ultrasound produced higher yield of phospholipids, especially the concentration of PI was relatively high. PI and PE was relatively high. The concentration of PI was around 31-fold increment compared to the untreated samples. This particular ingredient is of more technological interest. The PI and the PE is used in the liposomal preparations. And moving on to the study 3. It's in order to evaluate the effect of temperature, the solvent ratio, and the time on the total phospholipids, we did a response methodology to detect. And in this case, time was taken as a response for the yield of the phospholipids. This was done because of the reversibility nature of the amine, and which is based on the Le-Chatelier's principle, and we were able to obtain that the model was significant for all 3 responses, yielding in these 3 different percentage of phospholipids, and all these percentage of phospholipids are on the weight basis for 3 different times, 3, 10, and 18 hours for the minimized solvent ratio 3. However, in this study, we were able to note that the temperature didn't have much influence on the extraction yield, and low temperatures had a very promising results, very good it was really good for the ultrasound, good for the switchable solvent extraction. Further investigation of these studies can be done by using like how this temperature affected the individual phospholipids. Maybe that could be some degradation or some isomerization reactions going on between when there is the equilibrium prevailing before and after the reaction. This could be further investigated, but then we came to know that the temperature didn't have much effect on the switchable solvent extraction. Further, in order to understand the mechanism why the switchable solvent extraction without the use of any kind of polar alcohol give a higher yield of phospholipids, we came up with various analytical measurements to study the mechanism. This study of mechanism could be also helpful in developing various extraction strategies for the protein matrices, and also it can be used in other uh, it can be moved to like other using this switchable solvents. We can also use this in various other agricultural byproducts to get the oils and etc. In case of the particle size, we were able to see that the reduction in the size of the phospholipids using the switchable solvent extraction. This showed that lower particle size favored the diffusion of the solvents and diffusion of the solvents, and this favored the extraction efficiency of the phospholipids. And this confocal laser scanning images, which was stained using an RD dope dye for the phospholipids, clearly showed that the disruption of the phospholipid membrane using the switch was extensive in case of the switchable solvent extraction. And also the scanning electron images reveal that the disruption of the membrane as well as the separation of the lipid and the protein network, and the diameter of protein extraction was more in case of the switchable solvent extraction. Further, protein pattern were determined using the gel electrophoresis, which showed that there were severe protein modifications in the switchable solvent extraction, and also the confocal laser scanning images of the proteins and fats, which shows that the entrapped protein and fats in the untreated samples, which was disrupted further in the switchable solvent extraction. Clearly it was depicted. So overall, this study shows the disruption of the protein membrane by switchable solvents from the which favors the mass transfer between milk phospholipids surrounding the solvents and improving the extraction of milk phospholipids. Uh, in order to conclude, I would like to come up like the extraction efficiency of the phospholipids varied on the type of the byproduct. However, we were able to get a remarkably high amount of phospholipids, and higher caustic intensity favored the phospholipid extraction, and the model was significant for all responses. The microstructure and the charge particle analysis, which supported this mechanism on the disruption of the protein matrix, saying that it was extensive in case of the switchable solvents in such a way that this switchable hydrophilicity solvent can be used as a protein destabilizing agent and can potentially pair with different solvents to shorten the extraction time. So this process overall shows great potential for overcoming substantial financial and logistic bottlenecks, and it also helps the dairy farmers and industries to generate income and the industries to utilize the byproducts more efficiently. In future, we will be working on scale up of this process, and then ingredient formulation and the functionality of the using these phospholipids, and the retention kinetics of the heat and the oxidation stability of the phospholipids. Uh, and finally, I would like to thank the Dairy Management INC and USDA for funding my research program, and also South Dakota State University and New Mexico State University for providing all the research facilities to carry out my research work. Thank you. And if there are like, since if you want to know more about the methodologies, you can refer to this paper which we wrote. And thank you. I'll be happy to answer some questions.
Thank you very much, Kaavya, for your presentation. Very nice presentation. We only have time for one question, very brief. Okay, well, we are running out of time, so we have to move to the next presentation. But if you have a question for Kaavya, please put in the chat the question, and I'm sure that Kaavya will answer your questions. So we're going
Yeah.
to